Hello!
Hi there.
Welcome to Microbiology Twitter Journal Club. Microbiology YouTube Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was growing skin and infecting it with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated and aimed at the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked as a research and integrity specialist, and I'm currently working as an editor for a scientific journal.
And every week we meet to talk about microbiology. Today is our news week where we do an overview of papers that we've seen in the news or other sources to try to find an article that we want to cover next week in more detail.
Yeah, and you can follow along with the papers we discuss on either week in our shared Zotero library, linked in the doobly-doo. And next week will be our deep dive week, where we look at one of the articles that we are cycling through today, and we'll
Hmm.
focus in on it. So make sure to subscribe to
Yeah.
satisfy your microbiology curiosities.
So you can follow along with any of the papers that we're going to cover in our Zotero in the doobly-doo below. Yeah, and we want to hear from you. So please comment and tweet us using the hashtag #MicroTWJC.
Yes. So I read off of Danny's script accidentally during that intro, so thank you for. Uh,
All good.
Whoops, but we're good. We don't have any editors, so we're just gonna roll with the mistakes live.
I
So,
don't know if anyone can tell like that.
but yeah, so
um,
we've got a selection of interesting papers this week. So let's start with one that really hits on something that really affects us, is the fact that there is
yeah,
so
stuff that we've been
much.
doing this whole time.
Yeah, there is this one's called "Analyzing the Vast Coronavirus Literature with CoronaCentral." Um,
Mm.
So, I mean, yeah, there's so many papers to keep track of, and this paper describes one of the tools that people have been using, and we've actually, I think, used it once or twice to try and keep a track of
Yeah.
papers.
Just to see what was new that's out there. I mean, there's so many different, like, I think this is true for literature search,
Mm,
right? Like, people use different search engines. Some people use Google Scholar, like, there's PubMed is a great place to go. You can, like, try to find reviews and then go through the citations on those reviews. You
mm.
can do news sites. I mean, that's sort of part of what we've been trying to respond to is, like, news stories and things. But of course, like, yeah, there's so many ways to find literature. So I had used this in the past to try to find literature. But I don't know if if it was that helpful necessarily. It seems like though this paper is saying that it's just nice, like it's a good thing for like archival, right? To sort of understand the breadth of stuff that's out there.
Yeah, I mean, it describes about how it uses natural language processing to try and find papers and select ones that might be interesting for people, and
Hmm.
it analyzes the main topics that people are publishing on, and also looks at, and this paper actually describes the main like places where those publications are happening. And it seems
Mm,
so. With one figure, kind of shows that there's a massive amount in the preprint literature. So
totally.
yeah,
And we've seen that, right?
yeah,
We've covered so many preprints. And then I think what's been interesting too as we've been doing this program is we see those preprints also show up then in the journal articles. And
yes,
sometimes they're exact lists, but sometimes they do change a little bit.
yeah,
Like the last week's news, right? I think that we saw that, some like supplemental figures get cut out and things. And I mean, it goes to show like, there's so much data behind these stories of the papers, and the papers are just like how you can connect all that data into this narrative. But that doesn't mean that, right? Like I think as scientists, you're trying to figure out new narratives to tell with the data that you have, right? And the new data that you bring to the table.
yeah. And also, like, kind of, it's interesting looking at the different things that get published in different areas. So we haven't actually touched on anything from archive, mostly because those seem to be forecasting and modeling papers, diagnostics. I mean, I know in my like day job where I work for an engineering journal, like we've seen a ton of papers on computer vision, looking
Mm.
at X-rays and other things for COVID-19, and there has been
Hmm.
this issue where you get an engineering technology and they figure some way to kind of lever in COVID-19 in the title so that they can try and get it, like free open access or other sorts of deals on it. So
Sure.
again, this doesn't necessarily talk about quality, but it's very interesting about what, because again, we're quite interested in molecular biology, so quite a lot of our stuff comes from bioarchive, but there's a lot of other
Yes.
things from meta. So it's quite interesting to get this big overview of where stuff is being published.
Hmm.
So yeah, I found that quite interesting. It's like if Google kind of like opened up the search engine and said, "Hey, this is how we're selecting stuff for you to find." So it's quite
Sure.
interesting how they've got this academic version of it. And
So
again, you can, yeah.
so is it it says like how the algorithm that the search algorithm works in this paper? I didn't.
So yeah, it does. So it's in the supplementary material. So
I see.
but it is scrolled away there. But they use something called BERT-based topic on article type protection. So it stands for bidirectional encoder representations from transformers. Lots
Huh.
of words I don't know the meaning of, but they do mean a lot for people who are interested in natural language processing.
Got you.
Uh,
That's really
so,
interesting. Yeah, I guess because right, this is I think like the reality of search, right, is that like
mm,
you need like the key terms, but how do you sift out the key terms without asking everybody to like, oh, first of all, everyone's going to input different key terms, right? So like,
yes.
so they're using some sort of algorithm to extract those. Yeah, interesting.
Yeah, I mean the things like synonyms and understanding when like a word relates to say the therapeutics field or the biochemistry field or diagnostics.
Yeah,
Sometimes like you get something applied to all three, and you have to look at the context of that user search to think, okay, what is are they interested in like a new diagnostic test, or are they interested in the molecular biology of the biomarker used in that diagnostic test? So
sure.
yeah, so that's why I found that quite interesting.
Yeah, cool
Next
find.
paper. Another one. So this one attracts me mostly because of this fancy. They've got like some very fancy uh, like kind of figures on it. So
Hmm.
this like looks at the real cost of the outbreak. So it takes data from different countries, and
Hmm.
instead of going directly from the coronavirus, they look at the excess death rates because
Mm.
not everybody gets a positive coronavirus test before like they die, and especially when in cases where the healthcare system is being overwhelmed.
Yep.
So you can get points where the actual coronavirus deaths are underrated.
Yeah, I think I saw like I think I saw a news article about this at some point. It was someone's favorite like way of showing COVID data from 2020. Yeah, because it's sort of it's trying to get at this fact that like there are reporting deficiencies, right? People
Yeah,
are yeah, like people are dying in their homes in some cases, right? And that doesn't and if they do pass away in that circumstance, then they wouldn't have triggered, right? Like the sort of
mm,
surveillance machinery of going to the hospital or like, yeah.
yeah, and that hasn't taken into account like kind of there have been like various accusations of political aspirations, meaning that they try certain areas undercount deliberately. Um,
Mm-hmm.
so this is a way to get around that.
Try to
So it
bypass
pulls.
it. Yeah.
So yeah, I think this is an interesting one to, and say they delve into things like by country, also looking at gender data. So, and also this is yet another excuse for New Zealand to flex on people because for some reason their excess death rate decreased during the coronavirus outbreak.
Yeah, well, I mean, I guess if they were like we hear a lot of things that they were very responsible with like figuring out how to distance themselves from each other, right, and stay indoors. So, I mean, I assume that part of that could also be driven by like infectious disease rates going down, like flu.
Yeah. I mean, I think we did get some data from. So we looked at a paper a couple, I think, months ago from New Zealand about the flu outbreaks and how those got completely extinguished during the New Zealand outbreak as
Mm-hmm.
well. So
Mm-hmm.
they have got a lot of other benefits from that lockdown, mortality wise, which
Yeah. That
is
I was just thinking that United States graph there, it actually looks a lot like the graph of COVID-19 case. Like you really see those three lumps that we've had
oh
with the
yeah,
infection. Yeah. It's interesting how
yeah,
some
yeah,
of these look a lot like, I mean, I guess that's the whole point, right? Like if it correlates to the peaks and valleys of infection rates, then like that's a good indication that that might be driving the phenomena.
yeah. Oh gosh, yeah. This is. I mean, there's so much that we can delve into there. So if you're curious about like say death rates for people in certain age groups, people of certain genders, I mean,
Hmm.
it's really quite interesting, especially looking at like on and it breaks things down by countries. So you can filter it down to look at
Yeah.
say, as you said, looking at the USA, and you can see the the, the three humps.
Hmm.
Um, so yeah,
Cool.
it's a really interesting resource.
Yeah. Interesting find.
Uh, so the link to this is in the comments. So if you want to go on and play around with this yourself, then feel free to. I think this is quite a very interesting thing to look at.
Hmm.
Um, Yeah, and so the next thing I'm going to talk about is still on coronavirus, but on the variants, because now the B1617 is in the UK, and it's
Mm.
been split out into three variants. So
So wait,
B616,
oh, you're gonna say, right? That's the variant that arose in the Indian outbreak.
yeah, that's right. I think when we talked about the Indian outbreak, one thing that we noticed was that there were quite. It wasn't just like one strain of B1617. This, guess strain is the wrong word. One variant.
Yeah,
There's lots
variant.
of different variants, and of those, people were quite panicked about the double mutant. So there's one or two variants that had two mutations that might have made it more resistant to antibodies.
Hmm.
And so you have like three of these strains that are now in the UK, but two of them have that double mutation, and they're not spreading very well at all. The one that is spreading a lot is this one called B16172, and so this is a graph from the technical briefing so the UK government publishes a technical briefing report on the new strains and variants, and
Yep.
this gives a rundown of all the new things that we need to look out for, like and and the
Yeah,
thing
so
that
this was like the same this is the same group that was publishing, right? Like when we first learned of the B117 variant, right?
yes.
It was the work of the the genomic surveillance that is done in the UK that was giving us these stories and this data.
Yeah, and the UK have been putting a lot of effort into trying to get lots of genomics done.
Mm,
And so this graph I want to pull out quickly is showing. So in pink is B117, um, and that was the dominant strain. That's the Kent variant that evolved in the UK, and it was
mm,
dominant in the UK until somewhere in mid-April. Suddenly we started getting variants come from India, and
mm,
the Indian strain, which is in deep purple, has expanded quite rapidly over the past month or past
hmm.
2 months. So
Yeah. So this
that's
is this is like remember like I love that we have been talking about this for a while because like you get to see those similarities in the type of data. This is similar to when we saw the D614G mutation, right?
yeah,
When there are sets of mutations that sort of I guess have a fitness advantage, then you see them take over in this way. But then again, like there's always that the question of whether or not that's driven by a founder effect or whether it's
yes,
driven by some actual biological relevance of that, yeah, of that strain,
yeah.
of
I
that
mean, in this
variant.
case, it's probably there is something about B1617 that might be more transmissible.
Hmm.
Uh, at the moment, it's too early to tell whether there's any like extra excess deaths associated with it, and
Mm.
also the speed in which it's taken over. Part of that we need to acknowledge is the fact that the actual
Oh,
numbers of cases are much lower. So in this bottom part of the graph, you can see that the number of actual like suspected cases has gone down. So it's going to spread a lot more faster because there are a lot less people getting infected at the moment in the UK. So
mm.
it'll take a lot less time for it to take up a
Well,
higher
that's
level of
I guess
population.
that's what I'm saying. Like I think that that could like that also like it's more acute that it could be very much founder based because
Yeah.
it could just be these little clusters of infection that are going really high, but like it's not spreading between clusters necessarily. So it's only like that single infection event that drove all of this. Versus
Exactly.
if there were a lot of people being infected, then like there would actually be competition between like you got infected with this one and versus this one, or like that plays more of a role. Anyways, that's at
Yeah.
least how I was thinking about it.
I think you're right because let's see. I'm going to scroll down quickly because they've got some nice little graphs to try and represent the actual areas in which these variants start popping up and
Mm,
appearing. So,
mm,
uh, here's a graph of a map of England showing
yep.
where like this variant has been popping up, and mostly in high population areas and also some areas in like the north of England.
Hmm.
Again, these are showing like that these aren't isn't it's not like a big overwhelming uptake. It's just that in certain areas it's popped up really high.
Yeah.
Um, so that's some, and so in those areas suddenly there's been a lot more vaccinations happening, a lot more surge testing to try and get things under control.
Yeah.
Uh, and
Yeah, I
speaking
feel
of
like this is very much this is our current like pandemic scenario, right? Where it's
hmm,
like the rates are a little bit lower everywhere, but there's still a lot of viruses in general in the world. So like as things crop up, it's about like those systems to ensure that those outbreaks are contained and don't continue to spread.
yeah, I mean, it's like with smallpox, where they did ring vaccinations, where towards the end, when everything was dying down,
Yeah,
they'd like literally vaccinate everyone who was in contact with a person who'd had smallpox, and then their
right.
contacts, and
Yes.
and just target it down. And that's where we're going to move to that phase where we're targeting down small outbreaks and isolating them and treating them
Hmm.
almost like you would amputate a limb, but so
Don't
yes,
say amputate. That sounds horrible.
yeah, that's horrible. Don't quote me on that, people. How? Yeah, I
Yeah,
mean,
no,
make
but
treat.
I see
Yeah,
I see what you're saying. It's like, yeah, it's about isolating the outbreaks, right? Like you see like the little red map in the use, or it's like the game Pandemic, actually, right?
yeah,
Like we've moved to the phase where it's resembling the board game. You send the people to the places where the cases are highest and you try to get the work done.
yeah. So, uh, and speaking of vaccines, that brings us to the next paper, which is looking at the effectiveness of COVID-19 vaccines against the B16172 variant,
Mm,
which is quite. This is a paper that let's see. So it tests out like it looks at people who've had the vaccination and looks at the uh, the chance that they have had the infection.
hmm.
And essentially, what they find from this is that the that 2 doses of the AstraZeneca vaccine were 60% effective against symptomatic disease from the B162 variant,
Mm,
uh, and the Pfizer vaccine was a was a bit more than that. Let
mm,
me see if I can pull but because the way the paper so okay right so 88% effective for the BioNTech vaccine um, and and it's also 93% effective against the Kent variant as well. So
mm.
again, those have looked very good, and again, AstraZeneca it's less effective. But the thing is that because of the way it was rolled out, AstraZeneca was rolled out much later than than the BioNTech vaccine.
Hmm.
So and I think as we've seen
Oh,
in some papers, immunity accrues over time. So it might be that they
yeah.
in
They
this
don't have the
sample they didn't
data
have
yet.
yeah,
Yeah,
but this is
because
something that they keep
I think the important thing to highlight maybe is that these are being done like after, right? These aren't set up ahead of time. These sort of
no.
analyses. These are sort of post hoc
Mm.
exploratory looks at things. Yeah, to try to get a sense of what's going on.
Yeah, I mean now we know that the AstraZeneca vaccine is going mostly to the older people as well. So that's another
Hmm.
thing to take into account because
Right.
young people are more likely to shake off the infection or getting the BioNTech. So it'll look
Yeah.
like that one of them is
Hmm.
more better
Yeah,
when
like
actually they're the
that's a
same.
confounding factor. Like if this was done as a randomized control study, then those things would be balanced hopefully at enrollment. But here you can't do that, right?
Yeah,
Like this is
you
just observing how things are happening. But I think
yeah
this is a great point because we have talked a little bit about this. I think on stream is just that there's a difference, right, between setting up a randomized control study and like observational things. But I mean, the information that comes from both it can be very valuable.
yes. And in cases like this, you can't set up a randomized control trial because you have to know that this variant was happening in advance, and that's just
Yeah.
if you have that knowledge, why do you waste time doing that rather than preventing it? So, uh,
Hmm.
but yes, there are different levels of information, and we can just there is there are some things we don't know, but I mean the important takeaway from this paper is that vaccines do work against this variant, and
Hmm.
we can use that to stop them from spreading, and
Yes,
that's
absolutely.
yeah, and I feel
That's the promising thing, right? That's probably the value that these sort of studies bring to the table is to inform like, so how should we approach this policy, right? Or how should we approach the policy towards vaccines and outbreak control?
yeah, and I guess the thing is about vaccines, I think we're gonna the next one I'm gonna look at is going to be quite an interesting thing because this is talking about vaccine breakthrough infections.
Oh, okay. Well, let's just pause for a second. Minimal wrote, seems to be a lot of results showing convergent with in-host evolution driving this rather than transmission. I think talking about the expansion of the variant there.
Hmm.
So I mean, I think that that's true. So I have read a little bit. I don't think I've selected any of those papers about whether or not convergent evolution is driving it, but you have to see that like because the variants are tagged with like the set of mutations that it is, like these are not like I don't think that data that we showed from the genomic surveillance is supporting anything about convergent evolution, right? Like you're only going to get the convergent part when you sort of choose specific mutations, right? And say like this mutation seems to be cropping up everywhere, and then that mutation's not in the base lineage, and then it ends up appearing in that lineage, right? Those are the instances where you're seeing convergent evolution.
So yeah, I mean, I feel like minimal does have a point in there are certain mutations that we are seeing coming up again, like the the E484Q or E484K, and there are like some things that are definitely will
Yes.
uh
That might
because
not be they might not be in the base variant that ends up like taking over, but then as the infection continues, you see that particular mutation appear, right? And like that gives the variants their point whatever, right? Like that that's like how they're name that's part of the naming convention, right? But it's
yeah,
hard to it wouldn't be right to say that the variants themselves are converging, right? Like you, usually when we talk about the
right.
convergent evolution, it's just about that point mutation, and we're saying like this is appearing over and over again, and it's appearing across these different variants. But usually the variants have already been fixed, like when we talk, at least when I talk about them, right? Like I don't know about the news, but like when I say a variant, like I'm thinking about like a lineage that has a set number of mutations, and every point, whatever at the end of it, is another part of the mutations, and that may make it more similar now to other variants, and that would be the idea of convergent evolution.
Yeah, I think I can see what you're saying there because I mean the variants themselves aren't going to be having evolving to one kind of perfect coronavirus to infect humans because there's always
Right.
going to be some diversity in there. But we are going
Mm-hmm.
to see mutations crop up. I mean, one of the things I looked at in the weekly reports is that B117 has evolved one of the mutations that we've been seeing in other mutants that could have
Yeah.
an effect on is. And yeah, nothing is static at the moment. And the
Yes.
thing is, like, these coronaviruses are also competing against each other.
Mm-hmm.
So,
Mm-hmm.
so again, it's not. I'm not sure whether they'll be settling on one unique gene code, but there will be some things that we'll be spotting more and more, I think, and
Yeah.
we'll learn about how it interacts. I
Yeah.
had a paper in the docket that I deleted, unfortunately, that was about like how these variants will actually affect ACE2 binding and whether that will change and how that will
Yeah,
affect it.
absolutely. I mean,
Um,
that was sort of before we had all the reports of the variants, right? We saw papers where people did like saturation mutagenesis of the
yeah,
spike protein and just like tested ACE2 binding. So, like I think some of the papers that were coming out should synthesize that it should. I mean, probably what you found, right? Synthesize that previous information about mutations and try to like update it with what we've now observed as well, right? Like that's
yeah.
another way of indicating some relevance to mutations.
And also, how will coronavirus adapt to a vaccinated population? Because we don't necessarily know what will happen there.
Yeah.
Will it? So there's a lot up in the air. But yeah, that's actually a very interesting point. So thank you for bringing that, Minimal. Always good to
Hmm.
hear from you. Um,
Okay, so we said we were going to go on and talk about something about vaccines. Is that the next thing?
yes, COVID-19 vaccine breakthrough infections. So one of the big things that people are worried about with variants how are whether they can actually break through vaccination, whether they can evade the immune system by evolving these mutations. Because a lot of these all these like vaccines were based on almost the original version of the coronavirus that came out of Wuhan a year
Yep.
ago. But now the
Hmm.
variants have drifted somewhat. So how will we know whether those variants are able to evade the immune system? Well, look at people who've been vaccinated who have ended up in hospital.
Hmm.
So that's essentially what we're seeing here. So
Yep.
this paper focuses on people who've had vaccinations but still get sick afterwards.
Yep.
Uh, so
And this
so
shouldn't be so surprising, right? Because if rates are
hmm,
really high, we know that the percentage, right? Like vaccines weren't they're not 100% effective, right? So this is what
right.
let's follow up on that percentage that is not the protected percentage, right? Let's see what's going on, yeah, in those.
Exactly. I mean, in the original studies, they're targeting for about 60% effectiveness, or even 95% effectiveness for the BioNTech studies. And in this study, they looked at 101 million people who in the US.
Whoa.
On the And so from that you'd expect there'd be about 6 million people with breakthrough infections.
Hmm.
I think that this paper does undercount them somewhat significantly because they only look at 10,000 reports of breakthrough infections.
Hmm.
Um, so that would suggest that the vaccines are like 99.999% effective, and I don't think that's necessarily true or a good thing that
Yeah.
we want to say that they're one layer of
Yeah.
safety.
Well, I mean,
They're a seatbelt.
there's right. And again, like this data that we're seeing here, like this is not set up to show us this efficacy percentage, right?
Mm.
Like, so there's many factors, and we already talked about, right? There's many factors where you might get sick, you might be vaccinated, you might get sick, but you never enter the healthcare system, right?
Yeah.
In which case you'd never be logged like this. And the outcomes of those, like, we don't know what the outcomes are, right? Like, I think this is the huge challenge of all science, right? Is
Mm.
that you never have the complete picture.
Yes. Part of the big challenge of science, and especially in dealing with a virus that we still don't know that much about,
Yeah,
is making decisions in the absence of all full data. Like being able to make decisions with being able to take into account uncertainty. That's the word I was
right,
looking for.
right.
Uncertainty and dealing with that, and looking at levels
Absolutely.
of uncertainty.
So the value of studying this is not to say like how well is our vaccine working, right? But rather to understand that corner case, like what can the people that do not get protection tell us about what's going on, and should we be concerned, right? Like how like how should we be modifying policy if this seems to be going down a path that could be problematic in some way?
Right. Again, so this looks at some of the preliminary data. Uh, so 30% of the breakthrough infections were asymptomatic. So essentially these tests come from people who went to hospital for any reason. It didn't have to be to do with coronavirus. Sometimes it's through something completely unrelated, and they'd get coronavirus tests just as part of their going to hospital, and they found
Mm-hmm.
like that in people who've been vaccinated, 30% just were asymptomatic, which is kind of what we want. Um,
Yep.
ten
Yeah, well,
percent
I mean,
were.
it complicates the view. Like, so, for example, I'll say, right, like a lot of the reopening and the mask mandates in the States have been predicated on the fact that transmission is like reduced by 90%, right? But
Mm,
you have to remember that those studies, they weren't done as randomized control studies either. Those were observationals on healthcare cohorts
mm,
or right, healthcare worker cohorts. And so I guess or maybe what I'm saying is, it's still possible. Like we don't truly know like what the transmission rate for asymptomatic is, and like hearing 30% of these people are asymptomatic, that seems pretty. That's a lot, right? That's a lot more mm, than I
yeah.
would think. I guess maybe because like it's one of these things. It's like if you get infected and you've already been vaccinated, asymptomatic, that seems to make them. I don't. I have no idea. Do vaccines impact symptoms? Right. We would think that they do because it reduces the severity, right,
Mm,
and that's driven by symptomatic sort of events. But I don't know, right? It could also be driven in the variance of the people who are getting it, right? Like maybe
yes.
people get the breakthrough infections not because of anything about the vaccine or the virus, but the person who got it, right, is somehow more susceptible. So then, does that make them more likely to go down the asymptomatic route or the serious route? Right? Like these are all the types of things that I think are very complicated to
Yeah.
uncover in real life, right? Like we've only had these glimpses through different types of observations.
Right.
Mhm.
Yeah. So I mean, one of the interesting things about this is what this actually says about variants. So they did tests
Mhm.
for the different SARS-CoV-2 variants, and what you'd be expecting if any of the variants had a way to evade the immune system, you'd expect that one or two of them would be more represented. That
That's right.
so you got
Hmm.
like a certain percentage of
Makes
variants in the
sense.
population, and if one of them was able to evade the immune system, you expect to find more of that in whatever sample you get of vaccine breakthrough infections. And the
Hmm.
weird thing is they didn't find that at all. They found
Hmm.
that the variants that broke through represented the population, and
Yeah.
it seemed to be more likely that it's just the way that whether the immunity took in the people who were vaccinated. So the median age of people who are dying in this breakthrough cohort it was 82 years old,
Yep.
and I think we already know from the previous vaccination experiments that they've done that that vaccines are they do help, but there are some cases where they just aren't always effective for people in
Yeah.
that age cohort.
Hmm.
Um,
Yeah, actually,
so
that's a really interesting observation. I like that. Like, yeah, you would expect to see that all the variants are, if there was an effect of the variants, right? It would be an unequal distribution of them in this cohort, but there
yeah,
isn't. Yeah. Hmm.
so this cohort they spotted the Kent variant, the Brazilian P1 variant, and the South African variant. And I know that the South African and Brazilian variant, particularly in those antibody neutralization assays, tend to seem like they're quite scary. But in this
So
cohort,
okay, I'm not super familiar now. When you keep saying Kent variant, is that off the B117 or is
yeah,
that
that's
off?
B117. So that's what we're
Okay,
calling the
gotcha.
B117 now because we keep call. So I'd best call it the UK variant. Um,
Okay, yeah. I know
yeah,
Kent
sorry.
is a place in the UK, but I'm just like, is that the B117? I'm not sure.
I always forget I'm talking to an international audience. I do apologize, but yes.
All good.
Yes, the UK variant is what it is. So B117 is a UK variant that's become dominant worldwide, but again, it's not hasn't been shown particularly to be very. So it's not very like it doesn't evade the immune system that much.
Hmm.
The ones that we're frightened of are the South African variant, because I think we showed some scary papers where like some people didn't have any neutralizing antibodies to it, uh,
Yes.
who were
Hmm.
vaccinated. But in this case, it seems that it hasn't got that advantage. In this real world study, that kind of advantage it would have doesn't seem to be there, which is
But
very
but
interesting.
then but then remember, like that's also biased by the fact that it depends on the rates of those variants in the United States.
Right.
Right? Like those rates aren't equal in this area. So
Yeah.
yeah. And I guess like it just goes back to show like it's hard to get a grasp of this. But yeah, that small amount of information is somewhat reassuring, I suppose.
Yes. Now, the next paper is also related to the vaccine. So, Heterologous prime boost COVID-19 vaccination initial reactogenicity data. So, this is an interesting
Mm.
study because this study is testing whether you can mix and match vaccines. So, if you've
Yeah.
had
Yeah.
an AstraZeneca vaccine, can you follow that up with a Pfizer vaccine? And what will that
Hmm.
mean for immunity? So, this is very very early data from that. They've got
Hmm.
and the interesting thing about this is they've got four groups. One's like got pure Pfizer vaccine, the other one's pure AstraZeneca, and the other two are mixing and matching. So one group gets the AstraZeneca first and Pfizer second. The other second group gets the Pfizer first and AstraZeneca second.
Hmm.
So another thing about this is it also means that they're doing a first head-to-head comparison of these two vaccines.
Oh,
Uh,
sure. Yeah, absolutely.
so
Interesting.
this is very very early data.
Yeah.
So it's essentially initial reactogenicity, so like side effects. Uh, so it seems that like you can mix these vaccines, and the only
Safely.
issue is that sometimes the side effects might be slightly more when they're mixed together.
Hmm.
But again, this is something I mostly posted because I want to like keep an eye on this and let people know that this exists.
Yes, I
Um,
know. I think that's a really good point because I'm thinking about my dad actually in Canada. I know that like he got the first dose of the AstraZeneca, but then I heard that the AstraZeneca supply is sort of running out. So I'm not sure if he'll end up getting that second dose.
yeah, well, I mean, then he can get the BioNTech vaccine, and we'll probably find out from this whether it actually. I mean, I don't see any reason why it shouldn't work to boost immunity.
Yeah,
Uh,
I know from my perspective as well, from the immunological side, right? Like the
hmm,
booster is the booster, right? You're seeing the antigen. As long as the antigen is similar enough, right? And
right.
has the common epitopes, it should be boosting the response to them.
Yeah. I mean,
But yeah,
I feel like if we can. So the Sputnik vaccine from Russia essentially does that just on its own. It's got two different vaccine vehicles,
that's
and
right. Hmm.
so so I feel like with this, it, I mean they've almost. I mean, if you believe the data, I feel like there's some people who are very kind of skeptical about it. But if you believe that data,
Hmm.
then actually this kind of the process has already been proven, and we just want to make sure it's proven in this situation as well.
Yep.
Yeah.
Cool. Yeah, that's a good find. I'm glad that someone put this study together and that it's gonna
Yeah.
happen. I'm excited to see the final results of it.
Yeah, and this one is antibody response to two-dose SARS-CoV-2 mRNA vaccine series in solid organ transplant recipients. So, uh, again, we most of the studies we're looking at has been in immunocompetent people, people with like healthy immune systems,
Hmm.
but that's not the whole population. We want to make sure that this vaccine works in people who might not have a full and healthy immune system, because usually they're the people who are the most vulnerable to the COVID virus.
Yes.
Uh,
Yes.
so this study looks into that and looks into people who are organ transplant recipients, and as part of an organ transplant, you need to take these drugs to knock down your immune system because you have an
Hmm.
organ from another person, your immune system might recognize that as a foreign invader, and start to destroy it. So people usually get given a cocktail of drugs to knock back the immune system. And the question
Yes.
is,
And they
can the
usually take those long term as well, right? Because
yeah,
it's like it's these factors that sit on the tissue that your immune system recognizes. So yeah, if you have an organ, like you're at a baseline because you're taking these immunosuppressant drugs, you're at a baseline, your immune system's different. It's being changed
mm,
by pharmaceutical intervention.
yeah, and at this case, it shows that, like, say on the first dose, there a lot of recipients didn't have detectable antibody, but on the second dose,
Hmm.
quite a lot more of them developed that detectable antibody. Um,
Yeah.
and they comment that the antibody levels were still very well below than what has been observed comparatively in immunocompetent vaccinees.
Hmm.
So
Hmm.
this basically brings up the story of, okay, we can vaccines still help people who are
Yes.
immunocompromised, but there you still need that extra levels of protection and extra levels of care.
Yeah,
Uh,
like it may not. And I think that this is the real challenge, right? Because we don't know how those things correlate to then like real efficacy, right? Because
right.
there's not that strong link between just antibody levels and this, but from the antibody levels, we can say that they work differently, right? So the vaccines are working a little bit differently in this scenario, not exactly how they are in others. So there's reason to be quite cautious, I think. I think that's the main takeaway is that if you are someone who's an immunosuppressed, and I'm sure you already know that, right? If you're an
Right.
immunosuppressed individual, like, yeah, there isn't the same protection that's coming through. And so without knowing more about those peculiarities, like the idea is that, yeah, people should still be very cautious. And I think this actually opens up like a very important issue about like what does it mean to reopen a city, right?
Mm.
Because you reopen a city and okay, yeah, like all these vaccinated people can intermingle, but what about the people who can't, right? Like where's their city? It's their city too, right? It's
Right.
their locality as well. And I think policies that just say like, well, you can't do things anymore, like that I think is quite unfortunate.
Yeah. So this is the thing where, like, even if so, if we don't, if you don't pursue an elimination strategy, there will be some people who have to still keep up those precautions, perceivably for the
Yes.
rest of their lives.
Exactly.
If we
Yeah.
don't,
So I think this is a really yeah, that's a great tie into I think right a news one of the articles that we picked up last time, right? Like there
yeah,
could be two ways of thinking about elimination of the virus. Reduce it down to levels where it's like manageable, but then that means that these vulnerable individuals will continue to be vulnerable for the entirety of that time versus actual elimination where you're saying we just don't want it anymore in any locality. That's going to be yeah, beneficial for everybody.
yeah. Um, so the next paper,
Okay.
um, I think this was recommended by Minimal. Vaccination boosts naturally enhanced neutralizing breadth of SARS-CoV-2 1 year after infection. Um,
Mm.
so this is relatively similar to something I think we looked at in previous episode, where again this is from the same group who have been looking into like the potential of like long-term SARS-CoV-2 infection in the gut and
Hmm.
that enhancing immune responses over time or having
Yep.
some effect on immune responses over time.
Yeah, changing changing
Changing.
it.
I think enhancing
Yeah.
is the wrong term because it might not. We don't know necessarily.
Yeah. Yeah, so like in the title they say enhanced neutralizing breadth, right?
Mm,
That all comes together. It's not enhanced by itself, right? It's like
right.
neutralizing breadth. It's describing something about the inventory of all the antibodies that are being made. It's saying, "Oh, now they have more different types of antibodies that neutralize."
But yeah, so this looks at a cohort of COVID-19 formerly infected individuals who, after a year after infection, and 41% of them received the mRNA vaccines. Uh, it drills down looking at the antibody levels and also uh, picks out antibodies to see how they neutralize. Um,
Hmm.
so this is I mean, what would have been interesting would have been if they looked at people who weren't or initially infected, if they had a comparison group of vaccinated
Yeah.
individuals who didn't have a previous infection, so we could see whether the breadth is decreased in those, or if there is still some sort of expansion in the breadth of that,
Hmm.
because I feel like they've been like saying the kind of this quiet part of maybe there's this. So they've been very very subtle about talking about it because I suspect that they want to be sure before they send out press
Yeah.
releases or do anything,
Well,
make.
well, because another way of thinking about this could be that people just get exposed multiple times
Yeah.
to the virus, and the virus has been changing quite a bit, right? Like if you think about what the circulating strain was, right, like a few months ago versus the circulating strain now, I mean, I don't know how uncareful people are, but it could be that people get uh, repeated exposure of different variants, and that is what drives a diversity of antibodies. I don't know. I'm just throwing out that there without
Yeah,
having
no,
read this
I
paper.
think that's an interesting thing to take into account.
Uh-huh.
So there's, and yeah, I think speaks to like again going back to the variants and how there's a lot of things we don't know and how we might be overestimating somewhat in terms of the actual kind of vaccine escape because we don't know
Mm,
we've got a few ideas about what constitutes vaccine escape for variants, but we're
yeah.
still building that picture up.
I don't know. I actually I wouldn't interpret this data on the vaccine escape axis. I think I'm just because like I think that the paper is focused on the immune system, right? Like
Right.
it's trying to tell us something about how the immune system is changing during infection, and so yeah. So I think like like the only reason I am trying to think about like it could be because you see variants is just because like that's something that's in the environment, right?
Mm.
Our environment is a changing environment of the type of virus that we might encounter. So that just lies as a like an alternate hypothesis to the idea that it's some sort of like inside the gut. Like I don't think that that's what they're saying here, right? Like that's not what they've drawn out to say. But I think I'm just being primed from our last discussion to bring in that line
Yeah,
that
I
line of
think
thought. Yeah.
yeah. Um, so again, this is a paper that there's a lot of depth here, and the
A
abstract
lot of
is
it's a lot of B cell
a
stuff,
lot of
right?
lot of B cell
It's
stuff.
like this is like diving into B cell biology. And I was saying this to a friend just yesterday, like there's so much that we're learning about the immune system on this, right? Like so if you think about people who study the immune system, people have their favorite virus model to help understand the immune system, right? Like arenaviruses is
Mm.
this way to study the immune system through the lens of a viral model. And it's sort of characterized in a way in some I think that what we're seeing in the immune literature now is that coronavirus is a new model of understanding the immune system. So as
Right.
we understand more about coronavirus life cycle and the way that it works, we're also going to get like the mirrored knowledge of how the immune system works because we're seeing it across all these individuals, right? Like this is human data, right,
Yeah.
that we're looking at. And so human data means variation in human like responses, which is learning about the human immune system. That's something that is an amazing thing to learn because we usually understand the immune system of mice and we're relating that into humans.
Yeah.
So
I mean,
um,
the
that's
immune.
how I'm interpreting these types of papers. Yeah.
I think you bring up a really important point because yeah, the immune system is as complicated as the human brain, but I mean,
Mm-hmm.
in terms of like connections and the way information transfers, so it's really interesting what we can learn from this. Uh,
Right. Like, what if, like, just you could sub in, let's just sub in, for example, this is how I'm thinking about it. Sub in for SARS-CoV-2, just some other long-lived and pathogen that sits in the gut, right? Like sub in polio,
yeah,
right? Let's sub in polio and say, could we have learned the same things if we had as many, right? Like these studies maybe could have been done if there were the tools at the time, the attention on this sort of thing, and yeah, and the desire to learn this stuff about the immune system during the polio outbreak. But like that's not the
yeah.
case. We're in the coronavirus outbreak, and so we're learning about the immune system through that.
I mean,
Um,
yeah. I mean, I know this is going to be sound like a messed up perspective, but the same sort of thing happened with the HIV outbreak, where we
mhm.
learned quite a lot of the immune system because the HIV was directly attacking it, and we
Yes,
had to
that's
find out
right.
solutions to deal with that.
Hmm.
I feel like there's something similar going to be happening with SARS-CoV-2. I mean, the amount of science that's been happening and the amount of like conversations that have been happening about microbiology. I feel like there's
Yep.
there's a lot of things going on, and
Yep.
keeping a track of it all is a
Yep.
job in itself. Um,
So I think it is important that we can interpret data like this as telling us something about the virus, but keep in mind that the primary goal I think is trying to tell us something about the immune system. And the
mm,
virus is almost just like right, it's being like layered on top as like the instigator. But again, that's having not really read this paper very deeply.
yeah,
I'm just thinking about like how to interpret this body of literature, right? All the stuff that we've been seeing about yeah, how the immune system responds to this virus. what is it telling us of the virus versus what is it telling us about our own immune system?
Yeah. I and I think I'm going to move us on to the next paper, which is a paper that essentially explores the idea of long COVID. Um,
Mm.
this comes from an analysis of electronic health records. I think we've looked at
Hmm.
this group before because they published a paper on clotting that was
Ah.
brilliant because
Hmm.
it upset me both in terms of does it does it make people too frightened about clotting or not frightened enough. So I think that kind of makes them good in a way. Like,
Yeah, sure. They're balancing that tension, right? Of like presenting the data in a way that suggests multiple paths moving
yeah,
forward.
but this I actually found quite interesting because they were essentially characterizing the neurological and psychiatric outcomes of people with COVID-19. So looking at what, when after a COVID infection, what do people kind of say is wrong? What are the issues? And
Hmm.
they like pick up on like intracranial hemorrhage and stroke also being like kind of aspects of COVID that might happen. Also so things like
Yeah,
neuromuscular disorders. That's something that has been kind of mentioned with long COVID.
yeah. There's a huge list here on in table two, right, of all the things that they're looking at, and yeah, like I think one of these things that we've said a couple times is like there's just not a lot of reports and focus on this, right? Like we've seen like a couple reports, but the more data that we can get, the better. And this seems like they've really dove deep into trying to figure out. So again, this is not going to give us the mechanism, like do we know why this is happening, right? It's more the description of this is happening and at what at different levels, right? Like which symptoms are we seeing more frequently or less frequently?
Yeah, and they compare it to other respiratory diseases like influenza
Mm.
and looking at whether there's a difference, whether there's something intrinsic to like the place it's attacking, or whether there's something else that coronavirus is doing. And it
Yes.
seems
Yes.
like there is something that's happening there in long COVID that is
Yeah.
worthy of exploration.
I'm not super surprised, right? Because we have seen those, like that tissue tropism does exist for tissues that are not respiratory tissues.
Yes.
Right? We've seen those mechanistic studies. I mean, in cell culture or in organoids, actually, we've seen a lot of organoid style studies like that. So yeah, I'm not surprised. I'm glad that this is getting fleshed out because also if we can identify the main symptoms that we should be looking for, that can also help develop better disease models, right? Because like you kind of disease models need a symptom to latch onto or some sort of thing that
Yeah.
you can measure and say like, oh, this is kind of like this that we see in humans, and so like yeah, this is an important piece of data for that sort of study.
Yeah, I mean, it's almost the first stage of characterizing disease. I mean, historically, before we had any of these fancy tools in microbiology, most of the things that people did was just characterize what kind of symptoms people were having and then classify
Hmm.
diseases but based on that. Uh,
Yeah.
and again, going back to your point about looking not just about what this tells us about the virus, but also about what the human factor here, I think it's quite
Hmm.
interesting for people who might be suffering from chronic fatigue syndrome or things that might be related to those sorts of threats. Because if we
Sure.
so yeah, so I feel like this is a story
Yeah.
that is worthy of follow up, and I think it's quite an important one, especially considering we have had so many people who've been infected with COVID who might be
Yeah.
suffering from these. There's,
Yeah, we want to see more mechanistic research about long COVID, right? What are the things that underpin that phenomena? Because we've seen a lot of data say that it is a phenomena,
yeah.
and it needs to be addressed through research. Yeah.
So yeah, and it's going to take a long time because psychology and psychiatry is very difficult to get something solid like that from. So
Hmm.
it's going to take a lot of effort, and I'll be very interested to see what the results are.
Hmm.
Next one, SARS-CoV-2 infects human adult donor eyes and human embryonic stem cell derived ocular epithelium.
Okay.
So
So yeah, it's interesting. It affects the eye. It's like I've never heard of like donor eyes being used, but this is ex vivo, right? That's
yeah,
the approach.
yeah, ex vivo. I don't think they literally had people sit in front of them and they spat in their eyes to see whether they get COVID, but I
But I guess in some ways it's like closing the loop on the mechanism in which people like the mechanism in which hand washing is supposed to prevent the spread of COVID, right? This is closing the loop on that and saying, if it is able to infect this particular mucous membrane, this is the reason why we should not be touching our eyes, right? We should be washing our hands because we might be bringing those pathogens to this particular surface.
Yeah, I mean, it's kind of been assumed that because people have been wearing face shields and other things
Yep.
to like various spit guards and goggles to try and stop that happening. I mean, this
Hmm.
is, I mean, I feel like the eye is a vulnerable site for many diseases. I mean,
Yep.
while during my PhD, I accidentally infected myself in the eye with some strep.
Oh no. Oh, it's
It,
possible.
well, it was very. I was doing a dissection and a bit of
Mm.
bone flew into my eye and
Mm.
and I wasn't. So yeah, wear goggles, people.
Yes.
But yeah,
um, yeah, so I guess this is again, this is maybe an extension of a tissue tropism sort of study, right? It's giving us an ex vivo condition so that we know that this is more physiologically relevant, building on the understanding that right there's probably receptor availability right in these areas, and that's what's driving it. And yeah, and it's a possible route of transmission.
yeah. So that's quite interesting. Okay, so next paper. This next paper has been hitting the news somewhat. So it's got a long title. Vaccine-induced COVID-19 mimicry syndrome. Splice reactions within the SARS-CoV-2 spike open reading frame result in spike protein variants that may cause thromboembolic events in patients immunized with vector-based vaccines.
Mm.
So I feel like the first part of this title, the part that talks about like different spike even reading frames, is the part that is most justifiable. The second part is a bit hand wave, but let me describe what this paper talks
Yeah.
about. So it talks about mainly the difference between like the AstraZeneca vaccine and the Pfizer BioNTech vaccine, and
Hmm.
so one of them is an mRNA vaccine. So when an mRNA goes into the cell, it gets translated into protein in
Hmm.
the cytoplasm outside of the nucleus.
Yep.
Whereas DNA-based vaccines like adenoviruses, they have to go into the nucleus to get transcribed and produce
Hmm.
mRNA, and that mRNA then goes out the nucleus and gets transcribed into protein. Uh,
Hmm.
what the what these researchers kind of have caught on to is the fact that in the nucleus there's lots of like various ways that mRNA gets spliced. So
Mm.
the
I
internal
see.
process can chop it up into small pieces before it goes outside the cell. And
Yep.
what's so the idea here is that they've kind of tested out and discovered that there is a difference that in the AstraZeneca vaccine, because of this, it can produce various different spike variants based on these splicing.
Yeah, that's interesting.
The the problem that's a very interesting finding, but the problem is that they don't really link it to thromboembolic events. They don't have any evidence saying that. I mean, I mean, any paper that so yeah, it doesn't seem to have taken into account what the actual etiology of that disease is, or how these
Correct.
spike variants
Right.
might lead to it,
Right.
or whether, and
Right.
they've kind of boldly said that okay, this is the cause without actually plugging that kind of like
Yeah.
taking that
There's
step of testing that.
Yeah, there's one figure and they don't do anything like they don't do any like we looked at a clotting paper, right? And we saw some of the biochemical tests that go into that, right?
Yeah,
Like if you're gonna make a statement about clotting, then you can just take out like all those different clotting factors. Blood is a big field. Take some of those clotting factors, put in the protein that you think is doing the deed, and seeing whether the clots come out.
yeah,
And yeah, the figure that they have here is just gels of the splicing event.
yeah. The splicing. I'm not saying the splice event isn't interesting. I'm just
Yeah,
wondering, like, well, it's quite bold of them to say this causes clots without testing or looking at it. I mean,
absolutely.
by the same logic, you'd say that these spike variants they cause cancer and make you fly. I mean, we don't have the same level of evidence, so it's been a bit
Yeah.
frustrating in that
I'm
aspect.
sure they say something in the discussion that tries to link it, but I think that yeah, you have a very this is a really important point that like also an important point about reading papers, right? The title does not always tell you the whole story.
Yeah,
You like to at least scroll through and see a figure that speaks to that. And then you can be like, okay, they did something along that lines to try to support it.
yeah.
Um
And if the main purpose of your paper is said in the discussion,
yeah,
it's the
maybe
discussion
more of
is
a perspective, maybe more of a
yeah.
perspective
The
than a
the
research
discussion
article.
is where you apologize for all the shortcomings of your paper, or where you kind of try to make your paper or puff up your paper as much as possible. I mean, and it's one that tends to not get peer reviewed as well because people
Mhm.
just don't read it because it has nothing. It doesn't have the data in it or anything like that. So I mean, I remember this.
It can be hard to criticize, right? Because it
Yeah,
also is a little bit like it's a little bit of a thesis, right? Saying that given my data and this other data that I've cherry picked from the literature, here's how I think everything works.
yeah. So again, it's kind of bold of these researchers to approach all these press or various or allow themselves to be exposed to the press when
Mm.
this hasn't
Hmm.
even been peer reviewed yet, and
Yep.
it doesn't
Yep.
necessarily say what they want it to say. It still has a very interesting story that I would like to explore more because again,
I
this could
know.
be interesting in terms of
Alternate
like
splicing.
yeah,
No,
alternates.
it's about vaccine technology. Actually,
Yeah,
that's what's hitting me at first, right? Because
yeah,
these vaccines, like this is a huge rollout of different vaccine platforms, right? That we've had studies on them, but we haven't had large scale use, and knowing that they're processed differently gives us ways of maybe making them better, thinking about the relative strengths and weaknesses, right, of the different platforms. We have no information around that, and this is like a great mechanistic finding to help us begin to develop and articulate those
yeah.
points.
And this paper they work on their kind of own variant of so they don't work on like any of the they've got their own homegrown adenovirus that they test on. Uh,
Yep.
so I mean, part of me wants to add to this defense. Well, maybe AstraZeneca thought about this when doing the vaccine and accounted for it, but then part of me thinks, is that really what they did? I'm not confident they did that. So,
I have no idea, man. Like,
yeah,
I mean, they had been working on this vaccine platform for a while, right?
yeah.
There's like, it's so much work to dive in and see like the past, but yeah, who knows?
Who knows? I mean, but yeah,
Not us, not us.
not us. But yeah, so yeah, that's. But there are more papers about this that are also investigating this question about what's the cause of the side effects. So I'm going to start off with some like kind of good news from this case report about
Mm,
what this group where they found someone who essentially like they got the vaccine and they started feeling symptoms, and
mhm,
the researchers immediately noticed that okay, they took themselves to hospital and the researchers said, okay, right, let's just give you some treatment, some of the suggested treatment. So IVIG and various other treatments, and
mhm,
and they tested this person. They were getting that vaccine side effect, but they'd reached them early enough that they could prevent it from causing any extensive damage. So they're
hmm.
able to successfully treat this. So the only
Yeah,
thing is,
because this indicates the fact that IVIG is working, which is a therapeutic for the adenovirus,
yes,
right? That kind of makes me think that like why like I don't understand, like that is actually counteracting, right? The point that they were trying to make in the previous one,
yeah,
where it's like, oh, it's something about like the mRNA that's doing it. Well, no, this could just be that like the adenovirus is causing it in some way.
yeah. So in this case, what happened was after 8 days after, so a few days afterwards, she had a headache, but then like 8 days after the vaccine, she noticed that there was some uh, random bruises that came up that she didn't know what the cause of, like some bleeding from the gums, and and so she was like, okay, well, I'm gonna go take myself to hospital, and I think that's a wise decision for anybody who's had a vaccine. Like,
Yep.
if you see something, if you suspect it, go and check up immediately. I mean,
Yeah.
I had the AstraZeneca vaccine, so this is something I've been kind of keeping an eye on,
Hmm.
because again,
Yeah.
uh, so uh, so yeah, that's some kind of good news that this vaccine side effect is treatable. You just have to be
Yep.
kind of aware of what to look out
Yep.
for.
But I think we also said this when we covered like the paper that had all the serious cases,
Mm,
right? The fact that like that was published means that doctors know now to look out for certain things, right? And
yeah,
if you are experiencing effects that you think are coming, like that's a good reason to reach out for healthcare.
yeah,
And also doctors are also considering this, right? Like you don't have to take it into your own hands either. Like that knowledge is in the hospitals.
mm,
So you don't like there shouldn't be that I think there shouldn't really be that fear around vaccines in terms of like, but what about those side effects? Because there have been reports now and like that knowledge is sitting inside of the healthcare system.
yeah. And that brings us to the next paper, which is a bit more in depth. So this looks at the actual the actual structure of the vaccine and and tries to propose a mechanism for how that structure can lead to activation of PF4, which is
Mm.
what causes this uh, th- thrombotic thrombocytopenia. So
Yep.
the title of the paper is Structure of Chadox 1/AZD-1222 reveals interactions with CAR and PF4 with implications for vaccine-induced immune thrombotic thrombocytopenia.
Well, here's that mechanism. This is the type of study, right? That's really linking.
This is what we were looking for in the previous paper.
Yeah.
So this essentially suggests that that the surface of the actual not coronavirus, the adenovirus vaccine is what is
Adenovirus.
causing this.
Hmm.
So you've got some various structures of the adenovirus, and they kind of pair that what the charge of the adenovirus is what can trigger this.
Interesting.
So they do like a fair bit of modeling, and yeah, this is a lot more in depth, and it kind of
Whoa.
also
Yeah, I'm just
explains.
looking at like some of the structure. There's all this like structural biology of viral particles.
Yeah, uh, and yeah, they do some comparisons with the various other adenoviruses as
Mm.
well to see how they elicit the effects. Um,
Mm-hmm.
so yeah, this is very interesting. But again, it's a preprint. It hasn't been peer reviewed yet. Uh, it might take a higher level of structural modeling intelligence for people to find out weaknesses in this, if there are any.
Yeah, yeah. They don't do the like they don't again they don't have the biochemistry here, or do they? Where they're looking at actual clotting given these particles.
That's an interesting.
Fiber knob. They look at binding to these receptors. Yeah, they don't really have like a
Yeah.
blood version. Yeah.
So yeah.
They didn't develop an assay that, to me, right, like if this is the if this is the hypothesis, then I would expect that maybe I'm very old school, right? Like I would expect that you develop like some sort of biochemical assay to then address that, right? So add those viral particles into a reconstituted mix. This is just how I think blood
Yeah.
is done, like into the reconstituted mix of the things in the blood that you think are reacting. Show that that creates something like clumping or whatever. Maybe it's not a full clot, right? Or maybe it's just a association of the two,
Mm.
right? And then pull them down or something, and then remove each component and make sure that it's dependent on those components.
But yeah, that's exactly. You take the thing that you think is causing the effect, see whether the effect
Yeah.
happens, and if it's not
Yes.
happening, then see what part of the effect might be. And yeah, so just pull out the chain of causation. That's how a lot of
Yeah.
like microbiology and
Yeah.
a lot of science works. Um,
Right. Right. But this is like, I mean, this is still valuable work, right? Like it says, like we've predicted the interaction,
mm,
right? And we're gonna show you all that work. But like, the next step is like, then like let's bring it into that experimental condition. Let's see it on the bench. Yeah. Because then you still have to jump into the animal and into the human, right? Like there's still all those jumps. So like the closer you get, the better. That's always what we're
yeah,
driving for, I think, when we look at papers.
I think that brings us to the end of the SARS-CoV-2 aspect of the show. Now we're going to be looking at just generally exciting things in the areas of microbiology. Um,
Yeah, yeah. Since we're opening it up, right? And we're not we don't want to get pigeonholed into just talking about SARS-CoV-2 all the time. And
yeah.
there's plenty of interesting work that's still going on out there. Yeah, the first one we have here is a refined mechanism of mycoplasma mobile gliding based on structure, ATPase activity, and sialic acid binding of machinery. So this mycoplasma is I've heard about it as like a infection in cell cultures, right?
Mm,
Like your cell culture got mycoplasma all over it. It's a very small cell, and I didn't realize. I guess it glides as its motility. There's many different ways of moving around, right? Some people have a flagella. Some cells have a flagella and they spin it around and they zoom. But in this case, it's a gliding motility, probably from pili, I think is what they're looking at. Well,
yeah.
actually, they don't. Yeah,
Um,
I actually don't think they know what it is, right? Like the title says it all. Like they've sort of um, uh, 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 narrowed it down to a specific enzymatic function that is required, right? It needs energy to move.
right.
And they just have these awesome images, I think, is like
Yeah.
one of the things that attracted me to the paper. It's like awesome images of these cells up close, the structure of them. So just we talk a lot about the bacteria, the membranes of organisms, and how that mediates their interaction with the outside, whether that be preventing things from getting out or like directly binding, right? Even in just the last one, we were talking about the envelope of the virus. And so, yeah, just understanding more about this structure is very interesting.
Yeah, and the way I've mostly heard about mycoplasma is through the minimal genome project, where they're trying to create
Oh,
a,
yes,
bact-
that's right.
a bacteria with the smallest amount of genes, because mycoplasma are very, very small. They've got a very small genome. So if they're doing something complicated like gliding motility, then it might be through like a very simplified and elegant structure. So
Mm.
from the perspective of someone who might be exploring,
Engineering.
yeah, engineering bacteria, this could be
I think
quite an interesting thing to find out more about.
I think that's a
Um,
really big trend that I think has come up now, that we've read a lot of papers outside of just what we're looking at, but there's a lot of microbiology that we're discovering these natural things, and the backdrop of the technology it supports is engineering things. Like, can we reconstitute this system? Can we control this system?
yeah. So there's a lot of like interesting
Yep.
molecular biology they do here to try and like understand how this molecular machines work within it to try and move it forward and glide it. And so
Yeah.
there's and
I think
this
I think I think that's what we want to say about it.
yeah, it's quite interesting from that perspective. Next paper is single cell atlas of Plasmodium falciparum transmission through the mosquito.
I think
Uh,
people say falciparum.
Falciparum. Sorry, yeah.
Yeah, but I remember, like, you were saying that, I think 2 episodes ago you were saying you're really into the malaria story, right? And I was like, I don't
Yeah,
know if I want to do a I don't know if I want to do a story about a vaccine, like a clinical trial necessarily. And then I saw
but
this, I'm like, whoa, what about this one? Like, here we have one where they're doing our single cell sequencing of a whole mosquito.
yeah, I feel like you selected this with me in mind because
I did. I did. I did.
it's got
I was
two,
like, because this is something I could get behind. Like, you know, so we know that people aren't aware, malaria it's transmitted through mosquitoes. So its life cycle is both in us, but also in a mosquito. Like, it has to like get eaten basically, and then find its way into the salivary glands of the mosquito, so that then the mosquito can spit it into our bodies.
yeah,
Um, so yeah, so in this paper they do this relatively brand spanking new technique of single cell sequencing to figure out which cells exactly, right, are being affected by by the parasite.
yeah, and the parasite itself has so it changes form throughout its infection. So it's almost
Hmm.
like the way like a tadpole turns into a frog. This
Yep.
malaria changes all sorts of different like kind of uh, like forms that look different under a microscope. So
Yes.
it goes from and it undergoes sexual reproduction as well. So
Yeah,
that
which is
is
just crazy, right? It's
yeah.
it's a whole life cycle, like a whole. I mean, they're complicated organisms, right? They're yeah. Are they multi? They're not multicellular. They're
No, they're
single cell.
single cellular, but they do a lot with
Yeah.
those single cells.
Mm-hmm. Mm-hmm.
And yeah, we tend to focus in on like what's happening in the human. This one focuses in on the mosquito, and they
Mm-hmm.
also like put all their results on this website called the malariacellatlas.org. So
Yes.
we can so you can like do all sorts of filters based on say what stage of
Ooh,
the
oh,
life cycle
cool.
is. Uh,
Yeah, that's awesome.
there's a whole bunch of like, and you can like focus down things. It's
Yeah. So you can
really
imagine how
interesting.
this helps researchers, right? It's a atlases. This is like a big part of research. People publish their atlas, right? But the idea is that there's so much going on in terms of interactions between those life cycles in these cells that like we don't know what to focus on. And having an atlas, having an inventory of all the things that are going on, help researchers like zone in and say, oh, this is a really interesting thing. Like, let's follow up on that. And this paper is the publication of that atlas. how
Yeah,
did how did they make the atlas, right? I think that that's part of it as well.
it's very interesting. I mean, especially if you're like looking for say vaccine candidates and you want to focus on different stages of the life cycle, this would be
Sure.
very interesting to look at what biomarkers are there, and also like kind of what the genetic diversity, because they even like focus on like say you're like an like one say like in different stages in different like environments. So I mean, if you're like an oocyte that's currently in like the salivary gland, are your genes operating differently than if you're in a different part of the body? There's a lot of like interesting things.
Oh yeah, I think I didn't read this closely enough, but yeah, they don't look at mosquito cells at all. It's only the malarial
Yeah,
cells. Yeah. But it's the fact that in the mosquito they undergo all these life stages, so that's why it's titled in the mosquito.
yeah. Uh, so that's
Okay.
quite a fascinating story to look into. Let's see what next paper is. Global meat metagenomic map of urban microbiomes and antimicrobial resistance.
Mm-hmm.
Yeah.
Yeah, so actually, this is a study that's based in New York City, I think, or the MetaSub Consortium. I know that one of the main labs is here in New York, and there was this like, I remember the first time the group published something and it hit the news, and it was like, you can find all these bacteria on the subway, even like this rare or whatever. I think they even said plague at one point.
I knew
Anyways, like.
it. I always suspected.
But anyways, like I think since then, I think people have been better at filtering stuff and like also talking about their results. Here they're comparing the microbiomes across different cities.
Yes.
And sort of giving the sense that like I mean, this shouldn't be a surprise for anyone who knows anything about the microbiome. It's incredibly specific, right? Like and it's actually even more confusing because what we sequence is we sequence ribosomal rRNA, which just
Mm.
tells you identity, but actually different bacteria could do the same thing. So like so it's a much more complicated story. It's not that every location has different microbiota and so does different things. Rather, it's just like a fingerprint, I think. That's the best way of thinking about these types of things. There's a unique fingerprint, and that fingerprint is influenced by the environment and sort of the winds of whatever bacteria are around in the environment. And I think the way that they've tried to add relevance into this study is to then say, well, what can that tell us about antimicrobial resistance, right? Because you can zoom in and just look at the antimicrobial resistance genes, right, and say how is that then varying between cities, right, and how does that speak to the fingerprint that's being established?
Yeah, and they always seem to be going to so they take the samples from public transport. So
Mm-hmm.
which is always very interesting because it confirms all my suspicions about London Underground and various
Dirty places.
dirty places. But also
Well, no,
interesting. Yeah.
you know what? It's like there's this hope, and this is like a little bit of a dream and a bit of a tangent, but
Mm,
like there's this. We always talk about healthy gut microbiome,
mm,
right? But what does the healthy microbiome of like the built environment look like,
mm,
right? And just like in the gut, right? There are organisms that live there that outcompete things that we don't want to grow there.
mm.
And I would assume that that could extend to these spaces. And these types of studies are really the basis of building that sort of understanding, right? And I think that's one of the ways that we can talk about it when they bring in antimicrobial resistance is we can start to say, well, are there things that outcompete that antimicrobial resistance, right? And then does that mean that we could make our environments less conducive to the sharing of antimicrobial resistance, and thus hopefully, like by extension, somehow lower those rates in hospitals? I don't know, right?
Mm.
But that's where I see some of the relevance of this type of research and how they're framing it. Yeah, is going down that path.
Yeah, they also try to bring in like ideas for like climate, population density, and various other things to look at
Mm-hmm.
like how these urban environments differ and whether like the microbes
Yes.
respond to that.
Mm-hmm.
And
Mm-hmm.
also, I kind of have this idea now that or maybe I have a London microbiome, and if I take a poop in the wrong place, people can identify where I've been, and like if I
Well,
travel,
yeah,
well,
no,
yeah.
it is true. I mean, we talked about this briefly. There was a researcher talking about how with all these databases of studying disease, right, with a really easy way of just sampling a little bit of someone's genome, right, they might be able to find out if you exist in one of those databases, right? And so then there was this big push to try to
Mm,
encrypt that data so that researchers can still use it to understand the genetic basis of disease, but nefarious actors can't use it to like find out who you are. And like I mean, this is at the very beginning of the microbiome explosion. I feel like I was like reading papers at that time, right? And some of the first papers that came out was about fingerprints, right? That like your keyboard has a microbiome that belongs to you. And if you use someone else's keyboard, you could figure it out. um,
Wow. Yeah. Uh, Deep
okay.
stuff. And next one is this is quite an impressive paper. One cell metabolic phenotyping and sequencing a soil microbiome by Raman activated gravity driven encapsulation. Brackets rage.
Yeah,
Um,
I don't know why the rage is kind of intense. It's like they're at a little more intense level than I'm used to thinking about a research tool. But yeah,
but
this is almost an extension of what we were talking about in the droplet sorting.
yeah,
But now like just going into it and getting bacteria from the environment and just like seeing each one by itself. What does it make? What's the contents of its genome? Yeah, that's this tool actually had been developed a little while back. And I just saw this on a news source. I guess they had a recent publication where they put it in practice. So this is
hmm,
very actually, this is almost a mirror then to the way that I was talking about that previous paper, right? Where it's like they developed the technology, but they used a model system. And now they're going to use like in here, they're going to use soil, something real that they can learn something from and see how well it does to sort of cut through the complexity.
yeah. I mean, this is interesting for because it's not just
Okay.
categorizing. Well, for me, sorry. I mean, interesting for me because it doesn't just sequence the soil, but also tries to get their function by looking at what they metabolize, and they tag
Yes.
metabolites using deuterium. So
Mm-hmm.
deuterium is
Mm-hmm.
like hydrogen but heavier. So hydrogen has like an electron and a photon. Deuterium has that plus a
Proton.
neutron. Uh,
Mm-hmm.
and they use Raman spectroscopy to try and to analyze the presence of deuterium in these samples. So if a
Yeah.
cell ingests if they add some like substrate into the soil, like I don't know, like oils or like various things that you want to like for
Yeah.
bioremediation, you can actually like pick up which soil bacteria are breaking it down. And usually, if you're breaking down something that's complicated, it's not just one bacteria doing it; it's lots of them. So you can actually potentially
Yeah.
trace all those metabolites through the various food chains in the soil. And
Yeah.
with this, you can actually pick out which bacteria are doing what, which is fascinating for me.
Absolutely. Absolutely. Yeah. I just realized that we probably got I get so we get excited forget to like try to contextualize it more about like why is it so exciting that they can separate out single bacteria, because previously the way people approach this is they just grind up everything.
Mm,
Right, so like this is this era of single cell, all this microfluidic stuff, right, is really giving us a type of resolution that we haven't had before. Because even in tissues, right, you grind up the whole tissue and you take the average of all the stuff and you're like, this is what's happening there. But biology is happening at the level of the cell, or at least like that's an important sort of complexity level in which biology takes place. So understanding what's happening at that single cell level is like this whole new world of like of stuff and understanding that is very relevant. Yeah, so that's why this technique is very exciting here.
yeah. So there's a lot of like
Okay.
interesting physics going on. Next one is uh, efficient dilution to extinction isolation of novel virus host model systems for fastidious heterotrophic bacteria.
I like that they say fastidious heterotrophic bacteria, because I guess there's this type of bacteria in the ocean, SAR-11, right? Or that's a clade.
Hmm.
It's a loose grouping of bacteria, but it is like thought to be the most abundant organism, right, in our oceans.
Yeah.
And it was only maybe a couple of years ago that people even were able to identify that it's there and then grow it at sort of very dilute levels. We were talking before, we were talking about like growing bacteria for a long time, like months at a time in large chambers. There was a similar setup to growing SAR-11,
Yeah.
where it's like a very dilute water condition. You kind of wait for a long time, and eventually you get some of these cells.
Yeah.
Um, but every living thing has a virus. Well,
Yeah.
I mean, maybe that's an assumption, but like more or less, I think it's borne out. Most living things have their own virus. So what's the virus for this most abundant organism in the ocean?
Yeah.
And this group helps us figure out what that is.
Yeah. I mean, I mean, this is thought to be the most abundant bacteria on the planet, and we didn't even know about it for hundreds. We only learned about it in the 90s because someone was able to detect its genome. But even then, it took ages to
Yes.
actually grow it in culture. I mean, it's called
Mm-hmm.
Pelagibacter ubique because it's everywhere. If you've been in the sea, odds are you've touched it, you've breathed it in, and nothing's happened because it basically dies very, very quickly if you don't know how to take care of it.
Yeah, yeah. It's not adapted for it seems very well adapted for where it's living, which is everywhere in the ocean, but not really
But
anywhere
because
else.
it's such a big part of the ocean, it's a big part of the ocean ecosystem, and it's a big niche for any virus to live
Yes.
in. And if a virus can kill off
Hmm.
those bacteria, then that actually releases lots of carbon into lots of like nutrients into. So
Yes. That's right. Yeah, so I actually that's a great point that you brought up, that like that insight, because that's where this is where microbiology touches like
mm,
geochemical, right? Like kind of global geochemical cycling, right? Because microbes make up so much
mm,
biomass, right? Because there's so much of it, and they're so small and kind of out of the way, they do these biological reactions in just all these little places, and that's really important for nutrient cycling because we are one ecosystem. They are I've always said one of the fascinations that I have with microbes is that like they are the biological film that
mm.
connects all of us, right? Like they are the link, right? In some ways, the link between all pieces of biology because they exist in this
Yeah,
continuum. Yeah, just like over all the surfaces. So yeah. Anyways, I thought it'd be really interesting to maybe dive in and learn about
yeah.
this
I mean,
virus.
I did like a semester at a marine research lab where we did try to grow up viruses, and you usually have like a plate, and you grow up a lawn of bacteria and add a virus to that. But when you have something like
Mhm.
Pelagicobacter, like something like SAR-11, where it's so difficult to, they have to like resort to
Yes.
lots of different interesting ways to try and figure out how to find the virus. Uh,
Yeah, yeah, yeah. No, because I mean, at the end of the day, they do have these EM images
mm,
of it, so they must have been able to somehow get it to some density, but probably through
yeah,
filtering, right? Is my guess. Yeah, they grow them. I guess they have a little image of what they did. They have they grow things in seawater, and then they, I guess, filter to get to it.
yes.
Enrichment. I can't read that. So, oh, it's just an
So
arrow. Try to read an arrow.
yeah. So yeah, that's our list of papers that we've been making our way through.
Yeah.
Now for choices.
What are we gonna
It's a
choose?
difficult choice. There's lots of great ones. Um,
Yeah. I almost want to choose a SARS-CoV-2 one because we haven't been around it, but I don't know if any of them do any of them stand out to you?
Let me see.
Um,
Which ones? So s-
I was gonna say the vaccine mimicry one, but then the problem is that like actually it's only that
it's.
one figure and it's just about the
I feel
splicing.
like we've said all we need to say about the vaccine mimicry one. It's
Yeah. But we have an opinion on it. I want to talk about like how does the splicing work? Like what is the splicing that happens there?
yeah.
Yeah, but
I think
it's only one figure. It's kind of short.
yeah. I I understand the. I mean, I was quite interested in the Car4 and PF4 one, but
Mm. Hmm.
but let me see. I think there might be other ones that let's see.
From the past.
Yeah,
Uh,
from the
from the
past maybe.
from the past, we don't have that much SARS-CoV-2 from the past. I kind of removed a bunch from the potentials as we've been
Yeah, no,
going
that's
on. Yeah, I have like I mean this is I don't know if you want to do a modeling one. There's immune life history, vaccination, and the dynamics of SARS-CoV-2 over the next 5 years. Trying to like
ooh.
predict what's going on.
Uh,
But
so
that's a modeling paper. I don't know how you're feeling up if you're feeling up for modeling again.
I'm I'm not feeling quite up for modeling at the moment. I just don't have
uh,
the body for it. Anyway, so personally, like I think you know that I'm up for the malaria paper, the
yeah. No,
mycoplasma paper as well.
okay. No, I'm up for the malaria paper too. I chose it because I'm like, it sounds like you want to talk about malaria. This is one that I can get behind. So
So yeah, I think that this is the because so yeah, this
that
is
that makes it easy. When we put papers on the docket, I'm thinking about the others. It's an easy choice.
yeah because I was going to be divided by like okay the and it was divided by this and the was it the Mycobacter one because it has
Hmm.
videos. So you got the two things that I'm most papers with videos and papers with single cell sequencing, and but yeah, I
Absolutely.
think single cell atlas of Plasmodium falciparum transmission. I think that's the one we really want to look one I really want to look at.
Yeah. let's do it. Let's
Yeah.
do it. So join us next week for our deep dive week, where we will cover a single cell atlas of Plasmodium falciparum transmission through the mosquito.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I 100% agree. You can reach out to us over Twitter with the hashtag #MicroTWJC. We both believe that peer review is a process and that we can all participate in it. So I hope that you had a good time listening to us ramble about microbiology. And if you think you have something to add, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content.